Hey guys, this is Kalyan Ram, and today I'm here to give a small presentation on the project that me and my friends Satish Kumar and Srikanth have been working on for a while. So our project title is Greedy Algorithm for Minimizing the Number of Primers in Multiple PCR Experiments. So the contents of our presentation that I'm going to discuss now goes like this. Introduction, we're going to discuss about the background use cases of the algorithm, compare it with other brute force algorithms, limitations and also challenges that we faced, real world improvements, Python implementation. Why did we use Python to build this project, and also output. And I'm also going to let you know the work completed and also the work pending and member roles, how we distributed the work in between us, and what is everybody's contribution into the project, project goals, method slides, and at last conclusion. So yeah, this is the abstract slide. This is the abstract of our project. PCR is a widely used method in molecular biology to amplify DNA. Designing primers for multiple DNA regions is expensive and time consuming. Our project uses a greedy algorithm to use very less amount of primer and map the primer to maximum possible DNA sequence in one go. Um, Model the entire idea is to model this as a set cover problem. The number of primers used is considered as a set, and the coverage is basically the amount of DNA that we cover in one go. That is considered as a cover, so it is called set cover problem. The algorithm chooses primer that cover the most uncovered sequences in one go. A Python GUI was also developed to simplify input-output interaction. Initial results show a reduction in primer count. Future improvements include mismatch tolerance, reverse primers, and also biological validations like GC content and melting temperature. Uh, this is the introduction. So PCR is widely used to amplify DNA sequences, but multiplex PCR, it amplifies multiple DNA targets in one go. Using many primers increases cost and complexity. Our goal is to basically minimize the primer using a greedy algorithm. Key tool that we've developed to interact with our algorithm is graphical user interface, which is Python based. It makes the approach accessible for biologists and researchers. Um, these are the project objectives. The main objective is to minimize the number of primers without sacrificing the target coverage area. That is the amount of DNA sequence that we cover in one go. To develop a greedy algorithm to pick the primers and maximize coverage. Build a GUI for easy input of DNA sequences and primer length. Make the PCR process more efficient and affordable at the same time. Create a foundation for future bioinformatics tool with more advanced features. So background, PCR technology is widely used in medical diagnostics, pathogen detection, genetic analysis. Primer design is a crucial part of PCR. Multiplex PCR increases complexity as it has to map the primer to multiple DNA targets in one go. So it requires smarter algorithms to optimize the primer sets. Um, Use cases of this algorithms are diagnostics, fast and accurate disease detection, pathogen profiling, identifies multiple bacteria or viruses simultaneously. Biodiversity studies amplify DNA from multiple species using fewer primers. Genetic testing analyze large gene panels with minimal primers. agriculture biotech detect genetically modified organisms, GMOs, efficiently. Um, Comparison, we're going to now compare our greedy algorithm with other methods. Brute force, it basically tries all combinations, accurate, but in at the same time it is too slow. Dynamic programming, it's smart but harder to scale. Machine learning is dependent. We have to have really good data sets to develop a machine learning model to do the same work for us. And it's not always explainable. Uh, Greedy algorithm, quick and intuitive, scales well, works well using simple logic in Python. That is we're gonna use now. That is what we're gonna use now. Uh, Limitations and challenges. Current version only allows exact sequence matches, doesn't yet check for melting temperature, GC content, etc. Greedy algorithm isn't always globally optimal, struggles with very large data sets. When it comes to very large data sets, it does struggle a bit. So, yeah, these are the real world problems. Melting temperature range, GC content limits, we've already discussed. Avoiding secondary structures like hairpins, add reverse primer generation, allow primers with 1-2 mismatches. All of these have to be tackled down. And also integrate with tools like Blast to verify specificity. Export results in CSV or FASTA for lab use. Mm, and these are the method slides. So yeah, problem formulation, it is modeled as a set cover problem, as I've said earlier. Each primer is a set that covers DNA targets, primer extraction, all possible k-mers, fixed length sequences are extracted from the DNA. Greedy algorithm iteratively selects the primer that covers most uncovered sequences, continues until all are covered. Python implementation uses dictionaries and sets. So GUI is built using Tinker. Mm, accepts user input and displays results. Output validation confirms efficiency by comparing primer count with naive methods. Um, Python implementation. Why did we use Python? Python was used because of its readable syntax, powerful libraries, easy GUI development with and backend uses logic to extract the primers, match them with sequences, select optimal set using greedy logic. So this is the code that I'll demo later on. Uh, so yeah, let's discuss about the work completed. So what all work is completed? Um, is we've researched primary redundancy problem, implemented greedy algorithm in Python, extracted primers and mapped them to DNA, built a GUI for user interaction, validated algorithm with test sequences, created presentation and documented code for future development. Uh, this is the work pending that we should complete in near future. So final testing on large and diverse data sets. As I've said earlier, the greedy algorithm does struggle a bit when it comes to handling large data sets. So we have to test its limit on how much it can push. Uh, Add checks for GC content and melting temperature. Allow mismatches in matching. Implement reverse primer suggestion. Export results to CSV or FASTA. Improve GUI and algorithm based on feedback. So we're planning to collect feedback from the users and then improve our GUI and also algorithm, final polishing of presentation, report writing. So yeah, these are the member roles. So myself, my role is to basically make the project interactive and user friendly using the GUI. The tasks that I've performed are to design and implement the GUI using Python, allow input of multiple DNA sequences, display selected primers clearly as output. I have to display the selected primers by the algorithm to map maximum amount of DNA in one go. I have to print those. I have to make sure that our GUI prints the primers that are selected in the process and ensure that GUI connects properly to backend logic, handle errors or edge cases. And these are the roles of my teammate Satish Kumar, and these are the roles of my other teammate Srikanth. So yeah, uh, I'll show a demo of our code later on, and yeah, this is the conclusion of our project. We successfully solved a real biological problem using algorithmic thinking, built a Python-based tool that reduces primer usage in multiplex PCR, made the tool interactive via GUI. Our work basically saves time and cost for the bioinformatics, reduces primer dimer risks, can be expanded with biological validations. With improvements, this can evolve into a complete bioinformatics tool for lab use. So let me now just show a demo of our code. So yeah, this is the code that needs to be developed more to make it even more interactive and also make the algorithm handle large data sets in a more efficient way. So this is the DNA sequence and the target that we've set, and primer length is set to 4. This is the algorithm. So a function to find the primers, which takes the sequences, targets, and the primer length that we've given in this file as the input. And then the code it returns the selected primers and covered targets. This is the main code. So we have to click run and it's already done. Yeah, it shows the selected primers and also the covered targets and returns the total primers used. So yeah, that's it. Uh, that's it for today, guys. Thank you.